Hello, my name is Shaimaa Bakr, and today I'm going to talk about identifying key multifunctional components shared by critical cancer and normal liver pathways via a method I developed called sparseGMM. First, I will cover this talk's agenda. We will go over the motivation for this work, followed by previous related work. Then I will present an overview of the sparseGMM method. And finally, I will present the results of our work and a conclusion of the presentation. For many diseases, there are significant genetic underpinnings that determine both the underlying pathology and potential targets for therapy. Cancer is an important example of such diseases. In cancer, genetic and epigenetic events in certain genes cause dysregulation of biological pathways, downstream changes in gene expression, and cell signaling in a manner that increases cell growth and proliferation. With the increasing availability of genomic and transcriptomic data, there is a need to develop suitable statistical models and machine learning algorithms to leverage these large-size multimodal datasets to make significant biological discoveries and extract clinically actionable information. Specifically, our goal is to create mechanistic models that allow us to understand how gene expression is influenced by genomic events and to identify cancer regulators and their targets. Gene regulatory networks are one class of tools that can be applied to genomic data to improve our understanding of systems biology and to uncover the molecular basis of diseases. Several approaches to learning gene regulatory networks exist, including graph and module-based methods. On the left, we see Arachne, one of the earliest examples of graph-based gene regulatory networks. On the right, we see Conexic, an early module-based tool that uses copy number variation data and gene expression data to learn regulatory relationships. Here's an example of a module-based method to gene regulatory network learning that was developed previously in our lab called Amaretto. In Amaretto, we integrate multiple levels of omics profiles in the first two steps to identify candidate driver genes. Namely, we integrate copy number variation, DNA methylation, and gene expression data. In the third step, Amaretto connects these driver genes with co-expressed target genes that they regulate in modules, using a combination of clustering and sparse linear regression. Such module-based methods suffer from an important limitation, as they do not allow probabilistic assignments of target genes to modules. In this work, we present sparseGMM, a novel module network approach that combines a Bayesian framework with co-expression patterns to learn modules and their corresponding sparse set of regulators. Our method overcomes limitations of non-probabilistic module-based approaches. Additionally, while previous probabilistic approaches have focused on the development of discriminative models, we developed a generative model, which, as we will see, offers more flexibility and allows us to better model the biology of disease. In the context of GRNs, we classify genes into one of two types: target genes and regulator genes. We split the gene expression matrix into two matrices, one for targets and the other for regulators. Just as with standard module-based methods, we use the target gene matrix to try to find target genes working together or co-expressed. We cluster them together into modules, and each centroid represents its module. This is similar to the k-means algorithm, and so this problem can be solved with the expectation maximization algorithm. The expectation maximization algorithm is an iterative method to find maximum likelihood or maximum a posteriori estimates of parameters in statistical models where the model depends on unobserved latent variables. The difference here is that we have a probabilistic assignment rather than hard assignment as with k-means, and this is essentially how Gaussian mixtures work. In sparseGMM, the link of regulators to modules occurs when we represent the centroids as a sparse linear combination of regulator genes. So, we need to find the set of regulator genes that best explains the centroid of the module and consequently best explains the expression of the module. Sparsity is required since the removal of the sparsity constraint will link the expression of each module to a high number of regulators. This will overfit the solution and is not useful in helping us find true regulators that can be considered as drug targets. So we develop an expectation maximization algorithm to obtain a maximum posteriori estimate of the model parameters. Here, X is a matrix of gene expression that contains target genes. G is a matrix of gene expression that contains regulator genes, with the same number of samples but a smaller number of genes. Our objective is to learn beta, which is a weight matrix or regulatory program, It links the regulator genes to modules of target genes. K is the number of modules. And then, as typical with latent variable mixture models, Z is a categorical variable. Z sub I is a latent indicator of the mixture component that generated gene I. The probability of target gene X sub I is formulated as we see on the screen. We need this to calculate the likelihood of the data to solve our estimation problem. We typically look at the likelihood function of our data. However, in our case with a latent variable model, and since the cluster assignment Z sub I are not observed, the expected likelihood is used instead. Our algorithm requires two steps that are repeated until convergence. In the expectation step, the conditional distribution of the Z variable given our current parameter estimates are computed. In the maximization step, the model parameters are learned for the current iteration. In our case, we have this form for beta, and we are able to estimate the regulatory program. Because of sparsity in the estimator, we expect numerical instability, so we perform an SVD decomposition and adjust the computation of the estimator to avoid instability. We applied our new method to GTEx data from healthy liver tissue, as well as hepatocellular carcinoma samples from TCGA. As you may have guessed, our algorithm is not deterministic, and so with different initializations, we can expect to obtain different results. To measure robustness of modules and to filter for robust modules, sparseGMM is run several times, generating multiple gene networks. Next, robust modules are screened by running a community detection algorithm. This groups together robust modules that are consistently discovered in every run. To functionally annotate the robust modules, we apply gene set enrichment analysis to MSigDB gene collections. Finally, to validate regulators of robust modules, we use publicly available perturbation experiments from the library of integrated network-based cellular signatures or links that identify experimental targets. We combine normal and cancer modules to uncover shared biology between two types of tissue. We discovered 72 communities, 22 of which were identified to be robust using community detection. In this table, we see robust communities with Jacquard index greater than or equal to 0.7 and their links validated regulators. The main pathway of each community was revealed through gene set enrichment analysis against MSigDB collections. We see that sparseGMM is able to uncover modules with key biological functions in normal liver and liver cancer with very high robustness reflected in the high Jacquard index values. As an example, the analysis of HCC lesions showed that the clotting system process is robustly regulated in HCC and is driven by important regulators such as hepatocyte nuclear factor 4 alpha. The target genes of this module are also evidently associated with blood coagulation function. F3 is a coagulation factor, SERPINC1 regulates the blood coagulation cascade, FGG is a component of fibrinogen, the most abundant protein in blood clots, kallikrein B1 is responsible for activation of blood coagulation. The heat map on the right shows the strong co-expression pattern of the liver coagulation module. Next, we use single-cell evaluation of immune function modules to demonstrate the robustness of these communities. Looking at immune modules from TCGA data, gene set results reveal a distinct T-cell process as well as a distinct myeloid process. The table at the top left shows the top enrichments for the T-cell and myeloid modules. We evaluated the expression of target genes in an independent single-cell dataset of CD4 positive immune cells from tumor tissue of liver cancer patients. The heat map shows the expression of T-cell module target genes on the left and that of myeloid module target genes on the right. The top rows represent lymphoid and T cells, while the bottom row represents myeloid cells. This result agrees with the gene module assignment of sparseGMM. The average expression of target genes in the myeloid community is shown on the top right figure in green, while the average expression of target genes of the T-cell module is shown on the top left figure in purple. Cell annotation using PangloA-DB markers show that the T-cell and myeloid cell populations agree with module average expression plots. The bottom figures show the number of genes expressed in each cell type compared to other immune cell types, demonstrating specificity of target genes to corresponding cell populations. Next, we discuss the concept of target gene entropy. This is one of the unique and very useful aspects of our method. Only a generative model will allow us to calculate the probability distribution of a target gene as shown on this slide. The computed probability distribution can be used to calculate entropy. In general, entropy is an information theory measure of uncertainty inherent to the probability distribution of a variable. For a discrete random variable, it has the form shown here. Thus, high entropy genes are target genes which have uncertainty in their assignment. Interestingly, we observed that there is higher entropy in gene assignments in TCGA cancer data than in GTEx normal tissue data. This is shown in the box plot on the right, reflecting heterogeneity of cancer tissue. High entropy can be used as a proxy of membership of multiple biological processes and can unveil hidden elements of crosstalk in cancer. Functional crosstalk is an interaction between two pathways belonging to different cell processes. In the context of cancer, this crosstalk between pathways represents compensation mechanisms by which a cancer cell can generate resistance to the blockage of a specific gene or pathway. As an example, we examined crosstalk between estrogen and p53 pathways and found two high entropy genes with significant roles in both pathways. First gene is an estrogen-regulated gene expressed in ER-alpha positive breast cancer and is known to promote cell proliferation. Previous studies show its involvement in p53 through phosphorylation of HPIP. The second is a protein that is capable of both forming a complex with a well-known p53 target and of regulating estrogen receptors. We also found other elements of crosstalk between important pathways such as p53 and NF-kappa B TNF-alpha. To conclude, sparseGMM is a Bayesian approach based on a generative model that combines module-based gene regulatory network learning with sparsity constraints. sparseGMM robustly captures important biological processes in liver cancer and normal liver, as well as shared processes that occur in both types of tissue. sparseGMM uncovers multifunctional genes with potential involvement in functional crosstalk that can serve as drug target candidates and improve our understanding of cancer biology. And finally, experimental work is key in validating findings of sparseGMM in HCC. I would like to thank my colleagues and collaborators whose contributions have been crucial in making this work successful. Thank you.